All right, we're going to start with a brief recap of what we want to remember from Monday, because I know it's a little crazy Monday, possibly also Tuesday, but not for us. So when we were talking about how we set up for water reabsorption. So let's begin thinking about that osmolarity. Let me take a few more seconds here. See some answers coming in slowly. Don't want to rush you, but we do want to talk things through. Okay, let's take a look. Okay, so let's go over this then. So when we were talking about the loop of Henle and this medullary osmotic gradient that we're setting up, right? We're talking about kidney. It's got a cortex, which is its outer shell. And then we have the medulla. And we're thinking about basically what's in our renal pyramids is mostly what we're thinking about when we're thinking about this region. Okay, so we've got our nephron. It's glomerulus, it's Bowman's capsule, that whole renal corpuscle, and then the proximal tubule sitting out in the cortex. Right, then we have the descending limb, at least of our juxtamedullary nephrons, which is the kind we've been talking about. So, and it is the only kind we'll be talking about here. So don't worry about different types, but we got this loop of Henle going down and in. That's the descending limb. And then we've got the ascending limb coming up and out. Then we have our distal tubule and then our collecting duct, ultimately heading out of the kidney, though it passes through some other regions. Okay, so when we're thinking about that osmotic gradient, right, we've got this lower osmolarity of like 300-ish up near the top. And then as we get deep down, deep, we get to like 1400. Okay. I guess I should have drawn this loop of Henle going all the way down. We have some of them that go all the way down to there, right? And what's happening here, right, is we have fluid moving along through the nephron. This descending limb of the loop of Henle is actually still permeable to water. So we can think about it a lot like other parts of the nephron, so not particularly specialized, right? So we start to see changes in the osmolarity of the filtrate as it's traveling through these regions. So it's going to start matching pretty much the region that it's going through. So that's the descending limb, is the down descending, right? And the ascending limb, we see that filtrate starting to drop in osmolarity, right? So it's coming from 1400 back towards 300. And the reason for this actually is not the movement of water. So this ascending limb is impermeable to water. So this isn't an osmosis thing in that ascending limb. What's happening, right, is that we're grabbing ions, right? We're grabbing solutes and we're pumping them into this tissue, right? So into the tissue of the kidney, into this medullary region. And the presence of those solutes here in the medulla of the kidney is what gives it that osmolarity. So in the deepest parts, like now down by the bottom of the turn, we can imagine one even deeper, right? You've got lots of salts, lots of ions in that fluid. So you can pump out more of them, right? So you get a high osmolarity there. And you have less and less and less left as you come up to the top, which is also part of why we would see a decrease here. It's just like kind of another way to think about why we see that gradient, how we see that gradient. Setting up this gradient is super important because it sets the stage for our regulated reabsorption in the distal tubule and then in that collecting duct as we dive back down. So that was setting ourselves up, right? We're setting ourselves up with those properties of the medulla because what we really want to understand is, okay, how do we control water reabsorption? Because this is going to have a huge impact on our lives because it's going to, sure, change the composition of our urine, change how frequently we pee, but it's also going to change our blood pressure. It's going to help us regulate blood pressure, hopefully. Okay. So we were looking at that medullary osmotic gradient, right? And we think of osmolarity, bigger numbers for osmolarity. We think of them as a stronger pull on water, right? So we're pulling on the water as we get deeper. If we are able to transport water across the walls of this region, which we can only do in the presence of ADH, then we remove more and more and more and more and more water as we go down. And so ADH was the hormone we were thinking of there as triggering the insertion of aquaporin 2 in the apical membrane so that water can get all the way across the cell. Okay. But we know that sometimes, like our baseline state for both the distal tubule and the collecting duct is going to be a situation where we're impermeable to water, right? So we don't often think about cells that are impermeable to water. Our imaginarily generic baseline cell has aquaporins and we're just like not mentioning them, okay? But in the nephron, it's really important that we do have substantial portions of this structure that are not permeable to water unless we do something about it. So that's what we were setting up to understand the action of antidiuretic hormone. I think, yeah. All right. So if we look at the cell level, right, the cellular reason for this difference, right, is we're looking here at something called a principal cell, but the region we're thinking about more generally is that late distal tubule or the collecting duct. So similar processes happening in both, right? So we always have our aquaporin 3 ready to go here on the basolateral membrane, right? So we can always let water escape from the cell into the plasma. But we've got a big question mark about this aquaporin 2, right? So this is the one that we maybe have, maybe don't have, depending on what's going on with our hormones. So in this image, what we're looking at is when we have our hormone, ADH, antidiuretic hormone, antidiuretic. So we're going to think like making you pee less, right? Decreasing urine output, urine volume. Right? The way this works, this hormone swimming along in your plasma, swimming along in your blood, hits a receptor here on that basolateral membrane, which triggers a whole signaling pathway inside the cell. Okay. The ultimate effect of which is the insertion of these aquaporin 2 molecules in that apical membrane. It's the presence of those aquaporin 2 molecules that allow water to go all the way into the cell, and then they can still get out through aquaporin 3. So we got our water coming out of our, basically at this point, it's pretty much urine, right? So coming out of that tubular fluid on its way to becoming urine, pulling it all the way back into your bloodstream, okay? So this is the regulated reabsorption of water. So this is what we mean by regulated, not energy use or not, right? That's just active passive transport. That was happening in the proximal tubule, that mass reabsorption. Okay. Regulated reabsorption here has to do with the need for these hormone signals to tell us, okay, we're going to reabsorb now. So I think the last slide we left off on was thinking about why do we have the ADH in the blood in the first place? The answer is basically that your brain has sensors for this stuff, okay? So we have our osmoreceptors in this oval T region of the brain. Okay? It's special. You can sample the blood because it kind of lacks a blood-brain barrier here, which is great because what we're doing is sampling the osmolarity of the blood. So osmolarity is going to be higher. There's lots of ions, lots of salts in the blood. It's going to be lower if you have pretty dilute, watery blood. Okay. So when it senses lots of salts, this is an indication that, okay, like maybe you don't have enough fluid here, right? So this osmoreceptor here is going to be triggering the release of ADH, right? So we're going to release ADH from the posterior pituitary, which was the part of the pituitary that we thought less about in our endocrine unit, right? So that was where we had our neurohormones. So we might think of ADH as a neurohormone because it's being released from cells in the hypothalamus, cell bodies in the hypothalamus that have extensions, axons, axon terminals in the posterior pituitary. There are other signals for ADH regulation, ADH release, and we'll be talking about the interplay of different things. But one of the big reasons, right, that we might want to reabsorb all this water is to help us if we have long-term, potentially like drops in blood pressure, right? Like a way we could fix a drop in blood pressure, a drop in MAP. You need to get blood going through the kidneys and do all this reabsorption stuff, right? So this is going to be a little bit slow. But if you have a drop in blood pressure, drop in blood volume, you pull more water into the blood, it's going to puff up. You've got more volume. You've got more pressure back in your circulatory system. Okay. So the reason this has a down arrow next to the baroreceptor activity is like the baroreceptors like fire fewer times per minute. You can think of it as when the pressure and volume in the circulatory system is lower. So here is one loop we might look at as we are thinking about what's going on with the kidney and this ADH, right? So say we've got this high osmolarity first in the plasma, right? Plasma is a type of extracellular fluid. So it's in the bloodstream, basically, heading up to your brain. Right? We've got these OVLT receptors. So we can think of this as a reaction basically in the hypothalamus, right? So we've got these osmoreceptors here in the hypothalamus. kicks off our hypothalamic cells. They're like, okay, got high osmolarity, our blood's super salty. We don't like that. We want to water it down. Okay. So they send off ADH to the kidney, right? So the posterior pituitary connected to the hypothalamus produces the ADH, goes into your bloodstream. That blood pumps around your body, goes through the heart. Some of that blood goes to the kidney. We know lots and lots of blood is going to the kidney, like every minute, right? We had just fluid going into the blood or going into the nephron was 125 milliliters per minute, right? The plasma going to the kidney in the first place is like over 600, right? So lots of blood going to the kidneys. So we got this ADH going to the kidneys. Okay. In response to this ADH, kidneys in those distal tubules and collecting ducts, they're going from this non-permeable state with respect to water to inserting those aquaporin 2 molecules so that water can get all the way through, right? So they have this change, which allows for water reabsorption. Okay. So when we have water reabsorption, right, that water is now in your plasma, it's in the blood, which means it's not in your urine anymore, right? So in terms of excretion, what actually leaves your body, you're going to urinate less, right? You're going to lose less water because we pulled it back into the blood, right? And overall, this means we're going to be conserving body water. We're going to be keeping all the water that we have in our body. We're going to keep it in our body. We're going to keep it in our bloodstream if we can. So hopefully, as we pull more and more water back into the bloodstream, we're going to turn off the signal, right? We've got negative feedback. So once that osmolarity of the blood is back where we like it to be, right, those osmoreceptors are going to stop responding, right, because we don't have a problem anymore. And that's going to turn this whole loop off. So we'd stop making ADH. So those distal tubules and collecting ducts will go back to being potentially impermeable to water. Okay. So from the perspective of how this links up to our blood volume, our blood pressure, right? So say you're sweating a ton, or you're hemorrhaging, or you have some other issue with blood volume, right? And you don't have enough blood, right? You don't have enough volume of blood. This means you're also going to have low pressure in the system. So you'll have a low mean arterial pressure. Okay. And this is something that we can react to. So we have our baroreceptors, so pressure receptors, volume receptors. Okay. And they're going to detect and respond. So they're sending a signal up to your brain, sending a signal to your hypothalamus. So the hypothalamus is then going to do the same thing it did in our previous flow chart, right? So it's going to tell the posterior pituitary to make a bunch of ADH, which means our kidneys are going to reabsorb a bunch of water, okay? Which means we're not urinating as much anymore, right? So we're not excreting as much water, meaning we're conserving our blood volume, hopefully keeping enough blood in our system, hopefully, as you continue to drink some water, produce some water metabolically, bringing that blood volume and blood pressure back up. This is an important job of the kidneys, is maintaining our blood volume and therefore also our blood pressure in terms of like our long-term regulation. Okay. So that was the effect on the blood pressure, right? So the effect on mean arterial pressure. But we might notice what's going on with ADH by measuring urine potentially. So we want to think about, okay, so we know we're trying to keep blood volume up. So what is urine going to look like in the case that we are secreting ADH? What is it going to look like when it's not secreting ADH? So we're gonna answer here. What's our baseline state? So if we don't have a signal going to the posterior pituitary, what would we expect the urine to look like? right? Well, let's take a look. Okay, cool. We both got low osmolarity. Great. It's going to be high in volume because it still contains all that water, right? So that high volume is linked to the low osmolarity, right? So we've got lots of water in there and comparatively less those solutes, lower solute concentration because of all that high water. So this is what we would expect if we're not secreting ADH. So we'd have the opposite when we are. We'd have a lower volume of urine, but higher in its osmolarity. So that brings us to our regulation. Okay. So when we are regulating the function of the nephron, the function of the kidney, we know that an important factor for how our kidney is functioning, how our nephron is functioning, how this whole system is functioning is how much blood is coming to the kidney, what's its pressure, how does that affect our Starling's forces? So how does that affect our glomerular filtration pressure? And then ultimately, how does that affect the glomerular filtration rate? So like how much fluid is going to go into the nephron? Okay. So usually that glomerular filtration rate gets auto-regulated, right? So we talked about the fact that we don't really want tons and tons of extra fluid going into the nephron if your blood pressure is going up. We know that we have this big regulation kind of plateau here, right? So for a wide range of mean arterial pressures, we're trying to keep that glomerular filtration rate at 125 milliliters per minute. So we can do that through our myogenic response, right? So when the walls of that afferent arteriole are getting pushed out by a lot of blood, which would tend to increase the glomerular capillary hydrostatic pressure, right? And natural reaction of smooth muscle is to clamp back down, right? So that was our myogenic response. We talked about our other processes. So we had our juxtaglomerular complex partially regulating our filtration barrier. We have these ways that we are regulating that glomerular filtration rate in its normal spot. Okay. But we do have ranges outside of this mean arterial pressure region, right? So we might have super low blood pressures or super high blood pressures, right? So there is a region past which we can't auto-regulate these pressures, okay? So how this relates to what we're doing right now, right? So if you have a drop in that mean arterial pressure super low, right? So less than 80 millimeters of mercury, right? We are going to see a change to that glomerular filtration rate, right? So this is the point at which we are going to see a drop in that rate. So when we see a drop in that glomerular filtration rate, it's because we don't have as much of that hydrostatic pressure in the glomerular capillary. It was one of our big forces for filtration, right? So we're not filtering as much water, right? So we see a decrease in the water being filtered, which means we see a decrease in the water leaving the system, right? So if you're not getting as much water into the nephron in the first place, you're not going to get as much water leaving the nephron, okay? So this is somewhere we would want to up reabsorption if we can, right? In our higher range, right? So up above 180, right? This is another place that we would see actual changes to that glomerular filtration rate, right? So we see an increase. So now we've got a lot of fluid coming into our nephron, right? So we have an increase in the capillary hydrostatic pressure here. So we're getting more water filtered. It's rushing through like a fire hose. So you're probably also going to see more water being excreted. This should not be happening. So this is only going to happen in pathological circumstances. But this means that you're really pushing the limits and exceeding the limits of what your body can handle ultimately, if you see these big changes kind of long term, then this is past what your body can handle, even with this regulated hormone ADH. Okay. So we're going to move on to what's going on with sodium and potassium. And the spoiler alert for sodium and potassium is that both sodium and potassium are linked together. So it's going to be very similar things happening for each of these. So we tend to focus more on sodium, which we are going to continue doing right now. Okay. So when we're thinking about sodium balance, right, we want to keep an expected amount of sodium in your body because sodium is super important for your cell function, right? We're using sodium gradients all over the place in all kinds of different cells to keep action potentials working, right? So now we know that it's helping our glucose reabsorption. This sodium gradient has all sorts of different roles in physiology. Okay. So we're trying to regulate how much sodium you have in your body. You can have problems with your sodium, right? You can have too much sodium. You can have too little sodium, right? So remembering emia is referring to the blood, right? So we're thinking about what's the condition of the sodium in the plasma, right? So too much can be hyper. And I like that they put Na in the middle here, right? So remember sodium, Na plus, it's an ion, right? So we're saying too much sodium in the blood, hypernatremia. The opposite, hyponatremia, is going to be too little sodium in your plasma. And either of these can be an issue. Very jealous of my mother. The doctor has put her on what I term her all tortilla chip diet, and it involves, she's been told to eat tons and tons of salt all the time. And I hope I get whatever she has. I mean, not really, but like, yeah. Okay, so these are conditions you might see in patients. What's the point of that? Okay. So sodium is going to be your primary solute in the extracellular fluid. Really important. At this point, we've been thinking about sodium as it relates to our osmotic pressures, right? So we've just talked about how we have that ascending limb of the loop of Henle, pumping solutes, pumping our ions. Big thing it was pumping was sodium, right? It's super critical for excitable cells. So excitable is lumping together like neurons and like muscle cells, including like your heart. Okay. So the way sodium works with regards to the kidney, it's freely filtered from the glomerulus into Bowman's capsule. So we don't have a barrier for sodium there, any different than we have for water or whatever else, right? So it's just all coming in that same concentration that it is in the plasma. And we saw that in our mass reabsorber in the proximal tubule, we've got a lot of reabsorption going on. So it should not be a surprise that sodium is one of the things that we're reabsorbing a ton of in the proximal tubule. And now as we pass into the distal tubule and collecting ducts at the end of our nephron, we're going to be doing some more reabsorption, this time regulated. In total, you reabsorb maybe like 70% of the sodium in your plasma, depending. Sodium never gets secreted, so this is all going one way. Gets filtered into the nephron, gets reabsorbed into our plasma, but we're never going to be pumping it from the plasma back into the nephron. So we should never end up excreting more sodium than we filtered into the nephron in the first place. But we are going to regulate how much of that sodium is getting reabsorbed. So just like we regulated reabsorption of water, we're going to regulate reabsorption of sodium. But now we have two new hormones. Okay. So we're going to be reabsorbing sodium using aldosterone. We're going to see another hormone, atrial natriuretic peptide, ANP, also working on the sodium concentration, although they don't work in exactly the same way. So we're going to first focus on the aldosterone bit. Okay. So in terms of what cells we're thinking about. So we're in the distal tubule and we're in the collecting duct. They happen to have different types of cells in those regions. So specifically, we'd call these cells principal cells, where we have this reabsorption of sodium happening. Okay. So this is going to be active reabsorption. Okay. And it's going to rely, first things first, on that sodium potassium pump that we've been looking at on the basolateral membrane. Okay. Cool. So we saw something like this before with fewer labels, which was slightly annoying, right? But remember, plasma, so our blood flow, okay? Basolateral membrane is the side of the cell that's facing that blood flow. Okay, so here's where we find our sodium potassium pump, right? So this is taking sodium out of the cell, using energy to put it into the peritubular fluid, and then ultimately this is going to sneak into your plasma, into the bloodstream. Okay. So sodium going to get pulled across that basolateral membrane using energy. Okay. Here we're looking at the proximal tubule. So in the proximal tubule, what we were thinking about was this basolateral membrane sodium potassium pump, setting up this nice sodium gradient that we could use for secondary active transport of other things. Really, before we were thinking about the glucose co-transport with sodium. So like glucose would be the X here, right? So sodium now has this nice gradient, right? Where we have high sodium relatively in the tubular fluid, right? Lower sodium here in the cell because we've been pumping it out. And then high sodium again in this peritubular fluid, right? Which is why we can passively flow into the plasma at the end here. And so any of you who took bio of the cell as your prereq will hopefully recognize that you did a big problem set on this basically. Okay, so this is what was happening in the proximal tubule. So we did some sodium reabsorption. Part of the point of doing this sodium reabsorption was in order to help us transport important things like glucose. Okay. So this is unregulated. It's active, requires energy. Don't see any hormones on this chart here, right? So unregulated. The distal tubule, this is not showing us the hormones yet, but in the distal tubule, we are ultimately going to have regulation of our sodium reabsorption. But we're going to see similar situation, right? So we still see this sodium potassium pump on that basolateral membrane, right? In this case, they've shown us some secondary active transport of chloride ions here. But when we are thinking about our principal cells, which are going to be on the following slides, what we want to notice is that in the distal tubule, right, one of the ways that we are moving sodium and potassium across the apical membrane now, it's not secondary active transport, but we're straight up just opening channels, right? So when we have a channel protein, it just opens here, right? So we open a channel and that ion is going to flow down its gradient, right? Or with its electrochemical driving force, if we want flashbacks to A&P 1, right? So this is different than what we have in the proximal tubule, and this is the piece that's going to get kind of regulated as we think about aldosterone. Right. I want you to predict first. So what do you think would happen to the sodium transport if we increase the number of the sodium potassium pumps that are on the basolateral membrane, and we also increase the number of sodium channels on the apical membrane? So we're going to increase our active transport on the bottom. We're going to open some channels on the top. Okay, take another second or two. Let's take a look. Okay, cool. So majority are with us, but what you've just done, you've predicted what's going to happen in the presence of aldosterone, right? So what we were saying here was that we're going to increase our sodium potassium transporters here, right? So sodium goes out, right? Potassium goes in, okay? So what we've just done is we've dropped down that sodium concentration in the cell. So if at the same time we put a bunch of sodium channels here, right? Now we're going to pull sodium into the cell and we're going to pump it out of the cell. So the overall effect is we're reabsorbing more sodium because we're creating this gradient and then using this gradient to pull sodium back into your bloodstream. Okay. That's what aldosterone does, right? So aldosterone increases your sodium reabsorption. It's a steroid hormone. It comes from your adrenal gland. So your adrenal gland sits right on top of the kidney. Specifically, it's coming from the adrenal cortex, so that outer shell of the adrenal gland. Okay. And it acts on the cells of the distal tubule and the collecting duct. So specifically, it acts on principal cells, but let's not worry too much about cell types here. Okay. So what it's doing, right, is just what I just asked you. So it increases the number of these sodium potassium pumps on the basolateral membrane. And then it increases a bunch of sodium channels on this apical membrane, right? So we can now pull sodium all the way through into the blood. When I say that sodium and potassium get regulated using similar kind of pathways, I'm going to pick a different color for the potassium, but we might notice, right? So sodium, potassium pump here pulled potassium into the cell, right? So if you open a potassium channel on this apical membrane, here's how we get potassium secretion, right? So sodium goes one way, potassium goes the other way. Aldosterone does both of these things. Now, when we're thinking about aldosterone, we have a more complicated pathway, I think, for its release. So our ultimate goal in this renin-angiotensin-aldosterone system, or RAS, right, is to release aldosterone from the adrenal cortex. Okay. But in order to get there, we've got a number of different steps. So first, what we're going to do is we're going to secrete renin. So renin is coming from this juxtaglomerular apparatus, right? So the juxtaglomerular apparatus is a mouthful, right? But it's next to the glomerulus, right? Think like juxtaposition, right? So it's next to the glomerulus, right? And this means we've got the ability to sense what's going on with this arteriole coming in, bless you, right? So we've got this afferent arteriole here, efferent arteriole going out there. And here we can see that we've got part of the nephron coming back up here. So this is basically the tail end of that ascending limb of our loop of Henle heading into the distal tubule. Okay. We've got some specialized cells here touching our arterioles. And we had talked before about specialized cells kind of related to this, touching the glomerulus. Okay. So this is where we are going to be secreting renin. So specifically, our granular cells are part of this whole complex, right? So our granular cells, you can see some like here. Different color again. Okay. So we got some granular cells here that you can see on this picture. kind of next to this afferent arteriole. Okay. So these are going to be secreting renin. Okay. They're going to secrete renin in response to sensing some things about that arteriole and about this distal portion of the nephron coming back past. Okay, and we'll talk about the triggers for the renin release in a second. But first, that's where the renin's coming from. Okay. So this is how we get renin in the blood. That's the R part of the RAS. Okay. And what renin does is it converts a molecule that's always in your blood, a factor in your blood produced by your liver. So think like our clotting factors, right? We have clotting factors that were inactive, didn't get triggered to form a clot until you have a cut. Okay. Same thing happening here. You've always got some angiotensinogen coming from your liver, swimming around in your blood. This inogen ending is a signal that it's inactive to you. Okay. When renin is being released, converts that into angiotensin 1. Okay. Angiotensin 1 and swims along. It bumps into these enzymes embedded in capillary walls. Okay. So these ACE enzymes, these angiotensin converting enzymes, right? And we also have some receptors related to this, but they're in the walls of the blood vessel, essentially. When angiotensin 1 bumps into them, the action of that enzyme is to turn it into angiotensin 2. Thankfully, unlike our clotting factors, these are numbered in order, right? Order of action. Okay. And it's that angiotensin 2 that comes into contact with the adrenal cortex and tells your adrenal cortex to release aldosterone. Right? So this is why we have RAS, right? So renin comes first, converts the angiotensinogen into angiotensin 1. Then we have that ACE enzyme. So we can put all of that in this angiotensin part, right? And then the ultimate effect is the release of aldosterone from our adrenal glands. Right. So here we just got it big again. Right. So here's our blood. Here's that angiotensinogen from the liver. Okay. Here's that renin coming from that juxtaglomerular complex. Right. Specifically from the granular cells, but right up there in the kidney. That renin is turning the angiotensinogen into angiotensin 1. The ACE enzymes on our endothelial cells turn it into angiotensin 2. Touches your adrenal gland, which then releases aldosterone, which is its own separate kind of hormone. Okay. This is our pathway here for ultimately creating aldosterone. This ultimate goal is to reabsorb more sodium. I want to think for a second about the angiotensin 2, because it's in this flow chart. Okay. So when we're thinking about the RAS system, right, we're thinking about the relationship between angiotensin 2 and that aldosterone from the adrenal cortex. So here's our RAS. Angiotensin 2, just so you know, does actually have some other systemic effects. So angiotensin 2 was one of our bullet points that we mostly ignored in some of our previous units. So one other important action of angiotensin 2, now that we've been properly introduced, it has the effect of vasoconstriction on systemic arterioles. So angiotensin 2 tells us to make aldosterone. Angiotensin 2 tells us to do vasoconstriction. Angiotensin 2 also tells us to produce ADH from the posterior pituitary. So this is like another kind of signal for that ADH production. beyond just those osmoreceptors. So it's going to affect our water reabsorption. And it affects the hypothalamic neurons and actually has an effect on your behavior. So angiotensin 2 makes you thirsty. Okay. So it has a number of different actions once we're releasing angiotensin 2. But all of these actions, the final effect is going to be to increase your blood pressure. Okay. So our trigger for that release of renin at the beginning of that RAS system, right? That's how we were making the angiotensin 2 by these domino steps, right, are going to be some signals that indicate some drops in blood pressure. We'll have them listed out, but this is how this is all kind of weaving together. The other thing that we're going to note from this flow chart here, right? Say we're just focusing on the aldosterone part, right? So aldosterone in response to this angiotensin 2, cool. We reabsorb sodium, cool. We're going to want to notice that now we actually have kind of like a 1-2 punch going on. All right, so what we've done, reabsorbing a bunch of sodium. Now our plasma is salty, right? Now our plasma has this high osmolarity because that's where the sodium goes, right? So the sodium got into your blood, high osmolarity in the plasma, right? We can sense plasma osmolarity in our brain, right? So aldosterone is ultimately also going to be a trigger for ADH secretion through this extra step of affecting the plasma osmolarity. So there'd be potentially a little bit of a time lag here, but the sodium reabsorption and the water reabsorption are going to be linked together by this relationship. So I can see that we're at time. So we will finish up with this hormone regulation stuff next week. I'll time it out and get a practice exam up for you this afternoon because I know we got an exam coming up soon. Gotta figure out exactly how far I think we'll get by the exam.